Welcome to this lecture on deep sequence and language models. Today, we will explore how ordered data in the form of sequences are modeled using modern machine learning techniques, with a particular focus on applications in healthcare, biology, and global health. A sequence is an ordered collection of objects. Order is the key idea. These objects can be letters forming words, words forming sentences, sentences forming clinical notes, and so on. But it can also be chemical elements bonded into the constituents of biological systems. Thus, biology is fundamentally sequential. Our DNA is a sequence of nucleotides. Proteins are sequences of amino acids. Enzymes are ordered arrangements of proteins. Similar logics apply to lipids, glycans, and other complex molecules. But it also applies to higher-level biological systems, such as cells, tissues, and organs, which can be understood as collections of molecules, proteins, organelles, and other biological components. In the central dogma of biology, DNA is first transcribed into RNA, which is then translated into proteins. Importantly, the function or meaning of these sequences emerges at different levels of abstraction. Individual nucleotides make so-called codons. Codons encode amino acids, and amino acids form proteins, which themselves can sometimes execute complex chemical functions. This hierarchy motivates sequence modeling across multiple biological scales. Electronic medical records are also sequential. Clinical notes, lab results, medications, and diagnoses appear in time-ordered trajectories that define patient phenotypes and outcomes. Modeling these sequences enables prediction, classification, and clinical decision support. The core tasks in sequence modeling is essentially to predict the next element of a sequence. This means, given a sequence so far, what comes next? For example, given the phrase, chest pain and a metallic taste can be caused by, can a model infer the most likely continuation? Over the years, there have been several approaches and techniques for solving often non-trivial sequence prediction tasks across a variety of contexts. One of the earliest approaches to do this used a so-called fixed window to analyze recent tokens and applied a nonlinear transformation to predict the next token. While simple, as you can see here, this approach often fails to capture long-range dependencies and broader context. Many predictions require information far beyond the immediate past. For example, deciding whether to reduce sodium or allergens depends on earlier context about hypertension versus allergies. Fixed windows cannot reliably model such dependencies. The second big idea in sequence modeling came with the concept of a bag of words, which is a modeling constraint that attempts to represent text in terms of the presence, absence, and counts of specific words from a permissible dictionary while ignoring the order of such words. As you can see in the example, every word in a finite dictionary gets assigned a position, or binary encoding in the vector representation. If a specific word in the dictionary exists in the phrase to be analyzed, it is assigned a 1 or a 0 if absent. This representation could also encode the number of occurrences per dictionary word if the vector is non-binary. While computationally convenient, this presence and frequency-based technique often loses crucial syntactic and semantic information that is not readily apparent from the bag of words representation. Sentences with opposite meanings, such as, the surgery was good, not bad at all, and the surgery was bad, not good at all, have the same bag of words representations. This, of course, is a problem. If one can have identical bag of words representations for two phrases with complete opposite meanings, there is a fundamental issue here. That said, under certain conditions, bag of words is a nice and computationally bounded approach. If the kinds of sequences one wants to detect have distinct dictionary distributions and are less subtle than the example I provided. That said, because in language, order matters and bag of words discards it, this is not an approach that is still used frequently nowadays. Another big idea in sequence modeling was the use of large fixed windows with one-hot encoded tokens. Each word in a finite dictionary gets assigned a binary representation as a high-dimensional binary vector. Then these binary representations get concatenated into a large vector that covers the large fixed window, which is in turn the size of the model input. This, of course, preserves order locally, but causes the input dimensionality to explode, as we lack parameter sharing, and there is no bound to the input size as we want to use larger and larger sequences. An effective sequence model must handle variable length inputs, capture long-term dependencies, encode patterns efficiently, and share parameters to control complexity. And this is when the idea of recurrence for sequence handling emerged in neural networks. Recurrent neural networks, or RNNs, address these needs we stated previously by introducing feedback. At each time step, the network processes the current input together with a hidden state that summarizes past information. Recurrence is essentially the coupling of the old states with the current states at every sequence time step using persistent, non-changing functions and parameters. An RNN cell maintains a hidden state that is updated through a nonlinear transformation of the current input and the previous state. The same parameters are reused at every step, enabling parameter sharing across time. The hyperbolic tangent, or tanh, is commonly used in RNNs because it produces both positive and negative values. Here you can see the properties of tanh in comparison with the softmax and non-ReLU activation functions. This is particularly useful in text and sentiment analysis, where polarity matters, such as in good versus bad text reviews. RNNs and RNNs can be configured as many-to-one models for classification tasks, such as sentiment analysis, or many-to-many models for generation tasks, such as text, audio, or video synthesis. Here you can see the computational graph for the process of generating a sequence using an RNN, and along with that, obtaining the loss by comparing the RNN output at every step with the expected sequence in a supervised learning way. Training RNNs involves backpropagation through time, where errors are propagated across multiple time steps. As I mentioned, the loss is typically obtained from the comparison of the RNN outputs with the sequence elements that were supposed to be predicted in an autoregressive or supervised learning way. This process can be very computationally intensive and prone to instability. Standard RNNs suffer from exploding and vanishing gradients, making it difficult to learn long-term dependencies. Poor gradient flow can cause the model to forget earlier information or become unstable. Because of this, two tricks or ideas on RNNs emerged. One was careful parameter initialization, which empirically mattered and changed model behavior related to exploding and vanishing gradients. The other, more clever idea was the introduction of the so-called gates, which could control the flow of information in a recurrent unit to better modulate its learning and prevent unstable behavior. Gated recurrent units and long short-term memory networks introduced gates that regulate information flow. These gates allow the model to forget irrelevant information, store useful signals, and maintain stable gradients. A standard recurrent unit in a vanilla recurrent neural network, RNNN, a gated recurrent unit, GRU, and a long short-term memory, LSTM, unit on all processed sequential data by maintaining a hidden state that carries information across time steps, but they differ mainly in how they control the flow and persistence of information, particularly to address the vanishing/exploding gradient problem that affects basic RNNs. In a vanilla RNN, the recurrent unit simply updates its hidden state at time t, using a nonlinear transformation of the current input xt and the previous hidden state ht, with the output yt being the nonlinear hyperbolic tangent transformation of the weighted sum of inputs and previous hidden state, along with a bias. This architecture is simple, but as we mentioned, it struggles to preserve long-term dependencies because gradients decay exponentially during backpropagation through time. LSTM units solve this by introducing a separate memory cell, CT, and three multiplicative gates: input gate, forget gate, and output gate, which regulate what information enters the cell, then what information is removed, and what portion of the cell state is exposed as the hidden state. This gated additive update mechanism allows gradients to flow more stably across many time steps, enabling the model to learn long-range dependencies. The GRUs are a streamlined variant of LSTMs that merge some of these mechanisms. They combine the cell and hidden state into a single state vector and use only two gates: update gate, which determines how much of the previous state to retain, and reset gate, which controls how much past information to ignore when computing the candidate state. This reduces parameter count and computational cost while still mitigating vanishing gradients. Conceptually, you can think of the progression as RNNs use simple recurrent memory, GRUs use adaptive memory mixing, and LSTMs use explicit long-term memory with fine-grained gating. Where LSTMs offer the most control but are heavier, GRUs offer a good efficiency performance trade-off, and vanilla RNNs are rarely used for long sequences due to their difficulty retaining information over time. An LSTM includes input, forget, store, update, and output gates. Together, these components enable robust learning of long-range dependencies through a sequence of coordinated steps that let the network decide what to keep, what to discard, and what to reveal at each time step. First, in the input stage, the unit receives the current input xt, the previous hidden state h of t-1, and the previous cell state c of t-1, which together provide the new observation plus the short and long-term context from earlier steps. Second, in the forget t stage, the forget gate computes values between 0 and 1 and using a sigmoid function to decide how much of each component of the old cell, state ce of t-1 should be retained or erased. Values near 1 mean keep this memory, while values near 0 mean forget it. Third, in the store stage, the input gate decides which new pieces of information are important enough to write into memory, while a candidate memory vector, usually created with a hyperbolic tangent transformation, proposes the actual content that could be stored. Fourth, in the update stage, the cell state is revised by combining the retained old memory from the forget gate with the selected new candidate information from the input gate, producing the new cell state ect. And this is the core memory update that gives LSTMs their ability to preserve long-range information. Fifth, in the output stage, the output gate determines which parts of the updated cell state should be exposed as the hidden state ht, usually by filtering a transformed version of ct. This hidden state then becomes both the output passed to the next layer or prediction step and the short-term context forwarded to the next time step. This gated structure of an LSTM looks complex, but if you notice the top horizontal connection, the cell state, it forms a mostly linear path through time. So during backpropagation, the gradient can pass from one time step to the previous with little change. The cell state is updated mainly by adding new information and scaling the previous memory with the forget gate, so the gradient is not repeatedly transformed by weights and nonlinearities. In contrast, in a vanilla RNN, the hidden state is repeatedly multiplied by weight matrices and nonlinear functions at each step, causing gradients to vanish or explode. Because the LSTM only includes this identity-like memory path, gradients can flow across many time steps more reliably, and training is less brittle. Bidirectional training of RNNs and LSTMs is possible, a process where sequences are fed in both directions, providing richer contextual representations. This is especially useful when the full sequence is available, such as in genetics and even clinical notes. LSTMs are widely used for sentiment analysis, patient trajectory prediction, and modeling sequences of clinical events, such as diagnosis, treatments, and outcomes. The main purpose of medical sentiment analysis using natural language processing, NLP, and machine learning is to understand opinions, emotions, or attitudes expressed in healthcare data, such as clinical notes or patient narratives. Medical sentiment analysis connects three major dimensions. First, it operates within various medical contexts, including treatment side effects, any diagnostic concerns, the emotional impact of illness and disease risk factors, the patient-physician relationship, and physician attitudes recorded in clinical notes. Second, it involves different types of sentiment analysis tasks, such as fine-grained sentiment classification, emotion detection, aspect-based analysis that evaluates sentiments toward specific topics, and multilingual analysis across languages. Third, it can be implemented using different classification approaches, including traditional sentiment analysis, which can be lexicon-based, or modern NLP and other machine learning methods. The framework ultimately links sentiments to medical entities, such as changes in health status or disease condition, improved or worsened, diagnostic certainty, the outcomes of medical procedures, and patient responses to medications, including helpful effects, lack of benefit, or adverse events. Together, these elements illustrate how sentiment analysis can extract clinically meaningful insights from medical text. Beyond recurrent neural networks, gated recurrent and long short-term memory networks, natural language processing transformers have emerged as the most successful architecture style in language analysis, replacing recurrence with attention mechanisms. By explicitly modeling relationships between all tokens, transformers efficiently capture long-range dependencies without sequential processing. GPT stands for Generative Pre-trained Transformer. These models are trained on massive text corpora and can perform a wide range of language tasks. Modern GPT models contain hundreds of billions of parameters. We will learn much more about transformers in future lectures, but if you are curious right now, I absolutely recommend these videos by Grant Sanderson from 3 Blue 1 Brown. For global health and edge deployment, smaller models, such as Zephyr's 7 billion parameter language model, are critical, as those are now possible to run locally on portable devices without continuous internet access. So please make sure to be aware of this kind of small language models in the context of healthcare, as I anticipate those will become critical in safe and scalable future implementations of AI for global health. Okay, so this wraps up our rundown on basics on deep sequence and language models. But before we go, I want to walk you through some cool research that leverages these kinds of techniques. First, turns out sequence models are also used in synthetic biology to predict the function of DNA and RNA sequences. Interestingly, convolutional neural networks can outperform RNNs in some biological sequence tasks by capturing local structural patterns. In this example, the capacity of different RNA sequences to self-hybridize and be used for protein synthesis can be estimated by analyzing the nucleotide sequence alone. Interestingly, these sequences can also be visualized as multidimensional contact maps, which are essentially images that can leverage other techniques that we have seen so far in this series of videos, such as CNNs. Sequence modeling is, of course, used to power all the incredible developments that are happening everywhere in large language models and coding agents. Time series data such as ECGs, glucose measurements, and audio signals are also sequences. RNNs, LSTMs, CNNs, and transformers can all be applied to these problems. In practice, models are often combined. CNNs can extract local features, while RNNs or transformer models can model long-term structure. Choosing the right architecture is often an empirical process. Great, so I hope you have enjoyed today's lecture. And remember, sequences are everywhere, and effective sequence modeling underpins modern AI developments for language, biology, and healthcare. So make sure you try these techniques and get a sense of the world of possibilities it could bring to your own practice and to the world at large. See you next time, and thank you for your attention.